Hello, everyone. It is my pleasure to welcome you today to Komen's MBC Impact Series event for February. My name is Casey Volpe, and I manage MBC initiatives here at Komen. The programming for this series is envisioned as a year-long conversation with all of you. So every month is a new topic with new information and speakers. Today, we'll be covering new treatments and approaches to triple-negative breast cancer, metastatic breast cancer, my apologies. And our featured speaker is Dr. Melinda Telli, and we have a lovely moderator, Linda Lydia, who is an MBC advocate. Before we begin, I just have to take a quick moment and thank our 2024 MBC Impact Series sponsors. So thank you to our premier sponsors, AstraZeneca, Gilead, and Merck, and our signature sponsors, Daiichi Sankyo. One more housekeeping note. While our speakers are talking, please feel free to post your questions in the Q&A feature in Zoom. It's at the bottom of your screen. We will get through as many as we can, but please remember that our speakers cannot provide individual medical advice and are only speaking to their experiences. So please try to keep your questions general. No individual medical questions. Towards the end of the programming, a survey link will be placed in the chat because your feedback is really important to us and helpful for future programming. What topics do you want to hear? Who do you want to hear from? It's all there. So please feel free to share. Now, I'm very excited you all are joining us for today's discussion. And to start, I'm going to introduce you to our fabulous moderator, Linda Lydia. A native of Tennessee, Ms. Lydia is a graduate of Tennessee State University in Nashville, Tennessee, where she majored in sociology. She worked for 12 years as a probation officer in Dallas County before joining the U.S. Peace Corps in Sierra Leone, West Africa. In Africa, she taught swamp rice production in English. Ms. Lydia has a long and involved history with the NAACP dating back to the turbulent 60s and an active member of the Tennessee State University College chapter. Since these days, she served as the chartering advisor for the Grand Prairie NAACP, Los Angeles, California, NAACP, ACTSO chair and youth advisor, Dallas youth advisor, Texas State NAACP youth advisor, state ACTSO, and secretary of the Texas State NAACP at the national level as the Region 6 representative to the National Youth Work Committee. Ms. Lydia, you gave me quite the tongue twister because you're just so accomplished, but I am so impressed with you every time I interact with you. Ms. Linda also volunteers with numerous community organizations, including coordinating volunteers for breast cancer, Build, a yearly half-time home built by breast cancer survivors, Komen Race for the Cure Advocacy Chair, Komen AAHEI Cancer Coalition, Southwest Transplant Alliance, League of Women Voters, American Red Cross, Methodist Bread Basket, and others. She's proud of her mission work, as she should be, and she also works with Feed My Starving Children, having done missionary trips to South Africa, Haiti, and Cuba. She serves on the Speaker Bureau of FMSC. In 2020, Linda founded the Pink Diamond Survivor of Cancer Incorporated to raise funds and awareness for people experiencing cancer who are uninsured and underinsured. This group has been a major fundraiser for Komen, Sisters Network, and Celebrating Life. She's a 28-year metastatic breast cancer survivor herself. Ms. Linda serves on numerous boards and commissions, not limited to what was just listed here, and is an active member of Concord Church Dallas. Ms. Linda, the floor is yours.
Yes. I am at this point a 30-year breast cancer metastatic triple negative survivor, or thriver, I'd like to say. I'm really honored to be serving as your moderator today. I think there's so much for us to learn and to gain from this conversation with Dr. Telli in regards to triple negative breast cancer and options for treatment. Dr. Telli is a professor of medicine in the Division of Medical Oncology at Stanford University School of Medicine in California, of course, and is the director of the breast cancer program at the Stanford Cancer Institute and associate director of the Stanford Women Cancer Center. She received her medical degree with distinction from George Washington University in DC. She's completed an internship and residency in internal medicine at Stanford University, and then stayed at Stanford to pursue fellowship training in medical oncology. Dr. Telli research focuses on the development of novel therapy for the treatment of breast cancer, with a focus on triple negative and hereditary cancer, as well as metastatic breast cancer. She is interested in study designs that best the application of novel biomarkers in the clinical setting. Dr. Telli has served on numerous American Society of Clinical Oncology, ASCO, committees, and is currently a Komen Scholar, co-chair of the ASCO Breast Cancer Guidelines Advisory Group, member of the National Comprehensive Cancer Network Breast Cancer Guideline Panel, and a member of the Journal of Clinical Oncology Editorial Board. She is very well vetted for this conversation today. Thank you for coming, Dr. Telli.
Thanks for having me. I'll share my slides. And here we go. Welcome, everyone. Thanks for joining us today for this conversation where we're going to discuss new treatment approaches in triple-negative breast cancer. So I think it's always important to take a look back. When I started out in this field, the only treatment that we had for metastatic triple-negative breast cancer was standard chemotherapy. We didn't have any targeted treatment options. And while we had our standard chemotherapy drugs, there really wasn't great data in terms of how we should best use those. So it feels like it's been a long time coming, but you know, the last decade, we have certainly seen a number of advances for metastatic triple-negative breast cancer. And I'm going to go over today sort of the data that brought us to this point, and then end with some new approaches that I think are quite promising. So if we go back to 2018, this was when we had the approval of both olaparib and talazoparib. These are oral PARP inhibitors for patients with triple-negative hereditary breast cancer. And we found that these drugs improve progression-free survival in the advanced breast cancer setting. 2020 was an important year. This was when we had the approval of pembrolizumab added to chemotherapy for the initial treatment of PD-L1 positive triple-negative breast cancer. And then 2021 and 2022, this was when we had some of the new antibody drug conjugates, sacituzumab govitecan in 2021, also known as Trodelvy, and then trastuzumab deruxtecan approved in 2022, also known as Enhertu, for patients with triple-negative breast cancer that's HER2 low. So if we look at our current treatment strategy for metastatic triple-negative breast cancer, I think we've really filled this out from where we were many years ago. And as initial treatment, patients are recommended to receive chemotherapy alone or chemotherapy with pembrolizumab if the tumor is PD-L1 positive. So it has to be positive for that marker, and that predicts benefit from the immunotherapy drug pembrolizumab. We have the PARP inhibitors for patients with germline BRCA1 or 2 mutations, which can be given in the first through fourth line. And it is a nice option in the first line because it's an oral non-chemotherapy targeted treatment. For patients who've had prior treatment and need a new treatment, this is where we have the antibody drug conjugates, the sacituzumab govitecan and trastuzumab deruxtecan coming in, and then we still have chemotherapy. I think there are a lot of current questions. We have fortunately seen PARP inhibitors move into the early stage setting, as well as pembrolizumab, the immunotherapy antibody. And so there are some questions that remain if a patient received those drugs in the curative setting and then have a recurrence, you know, what do we do with those drugs in the recurrent setting? So that's sort of an area where we have a lack of data. And then when it comes to the antibody drug conjugates, the sacituzumab and the trastuzumab deruxtecan, what happens when we use these drugs one after another? Do we still see the same magnitude of benefit? And so those are some of the questions we're trying to work out. So I want to kind of talk you through how we got to this point. And more than, almost 25 years ago now, this was a really important project piece of work that came out of Stanford, along with some doctors in Norway. But basically, this was a very famous paper that looked at tumors and their gene expression and sorted these tumors into intrinsic subtypes. And there was one type of breast cancer that was described, this basal-like subtype, that tended to be triple negative, that had a very distinct sort of pattern of gene expression. And it was around 2005, if you look at the medical literature, this is when the term triple negative breast cancer first came into use. So we really kind of thought about these basal-like or triple negative tumors differently since that time, so about 20 years. And early on, a number of studies were undertaken really to sort of try and describe this group of cancers. And we found that if you look at triple negative breast cancers compared to others, triple negative tumors, they have a higher risk of early recurrence. And when they do recur, they're more likely to involve organ sites as opposed to non-triple negative breast cancer. We also learned a lot about the epidemiology, that triple negative breast cancers were more common in young women and more common in women of African ancestry. So we started to describe the features of this type of breast cancer. And one of the early observations that I think was important ultimately clinically is when they were doing these studies looking at gene expression in these basal-like tumors, which were enriched for triple negative breast cancer, they found that in patients who had inherited BRCA1 mutations, they tended to be triple negative and basal-like. And so this was sort of interesting that these BRCA-related cancers tended to cluster with triple negative and basal-like tumors. And this was important because a few years later, in 2005, this was when some of the very early laboratory work was done, showing that cells that had deficient BRCA1 or BRCA2 were very, very sensitive to inhibiting this PARP enzyme. And so this was the data that really propelled forward the clinical development of PARP inhibitors as cancer treatment. And as we marched forward, initial studies were done in the 2000s, and then by around 2010, some of the early studies were coming out. But we moved from phase 1 to phase 2 to phase 3. And ultimately, with these two trials, Olympiad and Embraca, we were able to get 2 PARP inhibitors FDA approved in 2018. And so we found that for patients who had triple negative breast cancer and a BRCA1 or 2 mutation, if they received a PARP inhibitor compared to standard chemotherapy, their progression-free survival, which is the amount of time that a patient lives with their cancer under control and not progressing, it was prolonged with the PARP inhibitor compared to standard chemotherapy. And then as we move forward, it was around 2010 or so that there started to be a lot of excitement around immunotherapy. And people started to look at this for patients with triple negative breast cancer. And there were some important papers around 2013, 2014, showing that if we look at triple negative tumors, they had, compared to other breast cancer types, more of these tumor infiltrating lymphocytes. So these are immune cells. So there are more immune cells present adjacent to triple negative tumors. And you can see at the bottom here that the more of these immune cells in your tumor, the better your outcome. So these were important for prognosis. And then there was another study done looking at this PD-L1 marker, finding that it was higher in triple negative cancers compared to non-triple negative cancers. And so this really paved the way to study immunotherapy, these PD-1 or PD-L1 inhibitors in triple negative breast cancer. And this moved from, again, phase 1 clinical testing to phase 2. And I'm showing here one of the really important phase 3 trials. This was Keynote 355 that took patients with their first recurrence of triple negative breast cancer, and patients were randomized to receive chemotherapy with this immunotherapy antibody, pembrolizumab or Keytruda, or a placebo with chemotherapy. And what we learned in this study is, unfortunately, the pembrolizumab didn't benefit all patients, but about 40% of the patients had this PD-L1 marker positive in their tumor. And in that group of patients, there was an improvement in survival, both progression-free survival and overall survival. And so there was an accelerated approval in 2020. And then by the summer of 2021, we had a full FDA approval for pembrolizumab for initial treatment of triple negative breast cancer. And with immunotherapy, if we look overall at side effects, pretty comparable between the two groups who received pembrolizumab with their chemotherapy compared to chemotherapy alone. Some slight differences, but in the overall kind of categories look pretty similar. But when we looked at these immune-mediated side effects or adverse events, there were certainly more of these in patients who received the pembrolizumab. And so some of these can be, rarely quite significant and require stopping the drug. I think most recently where all the action has been in breast cancer in general, not just in triple negative, but across all of the subtypes, has been with these antibody drug conjugates, or ADCs as they're referred to. And the first one that we had is this drug sacituzumab govitecan, also known as Trodelvy. And so this drug, it's an antibody, and it targets a marker that's very common on triple negative and other breast cancers called TROP2. So attached to that antibody are these, in red here, little chemotherapy molecules. And so the idea is that the drug is given, the antibody helps to direct this chemotherapy to the site of the tumor where the chemotherapy is released. So the effect is that you get more drug delivered directly to the site of the tumor compared to if you just gave that drug IV. And so this was the first big trial. They had early data with sacituzumab govitecan from phase 1 and phase 2 trials that there appeared to be a significant benefit with this drug for patients who had previously received chemotherapy for their recurrent triple negative breast cancer. But this was the phase 3 trial called ASCENT. And in this study, patients were previously treated with up to 2 prior chemotherapies, and they could come into the study and receive the sacituzumab govitecan or standard chemotherapy of physician's choice. And what we saw with this was that the patients who received here in green the sacituzumab govitecan, they did much better compared to chemotherapy, both in terms of their progression-free survival and overall survival. So there was a significant gain in overall survival with this drug. And this led the FDA to approve the drug for recurrent triple negative breast cancer patients who've received at least 2 prior treatments. And one of them had to be for advanced breast cancer. So this was really important because it was really the first new agent that came in showing good activity in a more heavily pretreated group of patients. The issue with the sacituzumab govitecan, while it is this antibody and chemotherapy drug combination medication, from a patient perspective, it very much still feels, unfortunately, like chemotherapy. You can see blood count lowering, diarrhea, nausea, hair loss with this drug. And so there are important side effects that need to be managed for patients receiving this medication. The next big antibody drug conjugate was this one, trastuzumab deruxtecan. Some people refer to it as TDXD. The brand name for this drug is Enhertu. And this was the DESTINY-Breast04 clinical trial. And so this is another one of these antibody drug conjugates. The antibody here, it attaches to HER2, and it is linked to these maroon-colored chemotherapy molecules. And this is sort of showing how it works. The antibody attaches to HER2 on the surface of a tumor cell. It gets internalized inside of the cancer cell, broken down, and then this releases the little chemotherapy molecules directly in the cancer cell. Some of these little molecules, though, can kind of spread throughout the cell and sometimes even cross the membrane of the cell into neighboring cells. And so the idea is that if this is a HER2 positive cell and this cell next to it isn't HER2 positive, there could still be benefit because there's this bystander effect where cells nearby can get affected by this chemotherapy. And so the DESTINY-Breast04 trial took patients who we have historically considered HER2 negative. But there are some tumors that they don't have the high level of HER2, the 3+ level, but they're not completely negative either. They sort of have some HER2. And again, historically, they've been considered HER2 negative, but this trial went out and said, well, can this medicine, the TDXD, show activity in this group of patients with either ER positive, HER2 negative, or triple negative breast cancer that has this low amount of HER2. And so for this study, patients had one or two prior treatments for recurrent breast cancer, and they were randomized to receive the T-DXd antibody drug conjugate or standard chemotherapy. And again, we saw, this is showing the triple negative data, improvements in progression-free survival and overall survival across the different subgroups in this study. And this led to the FDA approval of trastuzumab deruxtecan for any breast cancer that was HER2 low. So you could be triple negative, HER2 low, or ER positive, HER2 negative, but with this HER2 low status. And we saw, this is for the triple negative group of patients, the response rate with the T-DXd was much better than what we saw here with standard chemotherapy. And so that has been another antibody drug conjugate option. There are many, many more that are in development. And so this is another one called datopotamab deruxtecan, or Dato-DXd. This drug just recently was approved last month for hormone receptor positive, HER2 negative advanced breast cancer. And there have been early data showing activity in triple negative breast cancer. This again is an antibody that targets this TROP2 protein, very commonly present on triple negative and other breast cancers. And so this study, TROPION-Breast02, we are waiting to see the data from this, but if it is positive, we may soon have another antibody drug conjugate approved with Dato-DXd for patients with triple negative breast cancer. So this is data that we're waiting for at this time. There are a couple of other big trials that are going to read out soon. One of them is ASCENT-03. So this one is looking at giving that sacituzumab govitecan antibody as initial treatment for recurrent triple negative breast cancer. And so this study is for patients whose tumors are PD-L1 negative. And then there is a companion study, this one, ASCENT-04, looking at patients who have their initial recurrence of triple negative breast cancer, and their tumors are PD-L1 positive. So this is looking at initial treatment with the antibody drug conjugate plus pembrolizumab, the immunotherapy antibody. So we're hoping that we're going to be seeing these results from ASCENT-03 and ASCENT-04 in the next, by the end of the year. There's another one, patritumab deruxtecan, which is targeting a different protein. This one is targeting HER3, but this one has shown activity across a number of different tumor types. And this is just showing that in breast cancer, it has activity across all of the different subtypes. So there are definitely studies moving forward with this drug, HER3-DXd. So lots of activity right now with these antibody drug conjugates in clinical trials. One of the challenges we have, we currently have 2 of these antibody drug conjugates approved. Soon we may have more. And one of the things that we need to understand is how do these drugs work if patients sequence from one to another? And so this is a very active area of research right now, trying to figure out the sequencing. There's some data from the real world. This was looking at patients Here it's in hormone receptor positive, HER2 negative breast cancer, but there's similar data for triple negative breast cancer, showing that if you received Enhertu and then you go on and get sacituzumab govitecan, your likelihood of benefit from receiving sacituzumab govitecan after Enhertu, it's less than if you receive the sacituzumab govitecan without any prior Enhertu treatment. So this is sort of, a picture that's emerging that you get the most benefit with the first antibody drug conjugates. And while there are patients who will go on and respond well to a second one in sequence, for the most part, most patients have a lower degree of benefit from their second antibody drug conjugate. So there are some studies going on right now. This is one trying to look at the sequence question where patients are coming in, they're getting one ADC and then crossing over if their cancer becomes active to an alternative antibody drug conjugate. And patients are donating their time and energy and also agreeing to get biopsies so that we can try to better understand resistance to these different drugs. So this is an important study that's going on now. And then the last little bit here, I just wanted to talk about a new class of drugs that I think the data is pretty early, but I think is very exciting. And these are these new bispecific antibodies. And they're antibodies that target two separate proteins. One is a protein called VEGF, and the other one is the PD-1, PD-L1. Those proteins important in immune response. So just at a meeting in Spain in the fall, there were initial data reported from early phase studies from with two of these drugs. The first one here, this is ivonescimab. And this was one of these bispecific antibodies combined with chemotherapy as initial treatment for triple negative breast cancer. And this one targets the VEGF and PD-1. So it's different from Keytruda, which just targets PD-1. This one has this additional, targets this additional protein. So in this trial, patients had initial recurrence of triple negative breast cancer and got this ivonescimab antibody with chemotherapy. And overall, in this trial, there was a high rate of response for the patients that were enrolled. So 72% of patients had an objective response, which looked very good. And for a number of patients, this was still pretty early, but they were able to successfully stay on the treatment for prolonged periods of time. And what I think is most exciting about this new drug is that where Keytruda only works in tumors with the high PD-L1, it appears that this antibody works in all tumors, irrespective of whether the PD-L1 marker is positive or negative. So right now we have immunotherapy as an option for only 40% of patients. The hope is that potentially something like this could extend that benefit to all patients with triple negative breast cancer. And overall, in terms of side effects, any drug that targets VEGF, we have a number of approved drugs that target VEGF. These can lead to some unique side effects regarding high blood pressure, protein in the urine. But overall, most of the side effects in the trial were related to the chemotherapy, and most of the side effects were either grade 1 or grade 2, so lower grade. And then there were no adverse events in the study that led to patients stopping permanently, and no side effects that led to death. So encouraging early data, both in terms of effectiveness and safety with ivonescimab. And then there's another trial very similar, looking at a drug BNT327. And so this one's from a company called BioNTech. We know about them, of course, from the COVID vaccine, but before they did the COVID vaccine, they've been a cancer company with a focus in triple negative breast cancer. And so they have one of these bispecific antibodies. It targets PD-L1 and VEGF. And they did a very similar study, chemotherapy, nab-paclitaxel or Abraxane, with this bispecific antibody as initial treatment for triple negative breast cancer. And they also saw here in the overall population, over 75% of patients had a response to this treatment. And again, the responses were seen across all of the subgroups in terms of the PD-L1, whether it was positive or negative. So I think these two studies, they're early, but are pretty consistent in terms of the benefits being seen across all patients with triple negative breast cancer. And this was the safety here. In this trial, about 5% of patients had to discontinue treatment due to side effects. Again, most of the side effects are being driven by the chemotherapy, but there are these immune-related side effects, as well as some of the side effects related to blocking that VEGF. You know, these are side effects that we need to pay attention with as this moves forward. So this BNT327 is currently in phase 2 testing for triple negative breast cancer. We have this study open, and there is a phase 3 trial that is rapidly moving forward and hopefully will be open by fall. And so just some concluding thoughts. I hope you take away today a sense that we have made real progress in triple negative breast cancer. And if we look at our new drug approvals, they've all been since 2018. And I think if we look ahead the next 5 to 10 years, many more will come. We talked a lot about the highlights here. Of course, there are always, you know, other questions that were asked, studies that were performed that ultimately didn't advance. But I think we always learn very valuable lessons from these studies as well. I think looking at triple negative breast cancer, this, you know, we've only described triple negative breast cancer 20 years ago. And I think by being able to describe this group of breast tumors, really focus on them, we were able to advance the field. So I think it's a great example of how an area of medicine can advance when we focus on it. And I think the other great thing about this story is that contributions came from all over the world, really highlighting the importance of collaboration in science. I'm going to stop here just because of time for questions. Thanks, everybody. Linda, you might be on mute.
Okay.
There
The
she.
first question I'm going to pose, I'm sorry, oops, hold on. Okay, great. The first question that was posed was about the availability of clinical trials for triple negative now.
Yeah, so like I mentioned, I think if we look back at kind of the last year or two, a lot of the clinical trials have focused on these antibody drug conjugates, and I think that still remains a really big area of focus. So some of the existing ones, looking at using them differently or using them in combination with immunotherapy. But this new sort of bispecific antibody class of drugs, I think these are sort of drugs that are, the early testing has just been completed. And I think these are going to really move forward quickly into bigger trials in the next 1 to 2 years.
Okay, well, that's good to hear.
Yes.
How long has the trial been going on? I know I attempted to get into your, I guess it was BMEN 673 in 2017. And that was actually before they even, I guess, said they didn't coin the phrase triple negative until what, 2021?
What, 2005?
2005, yeah. Yeah.
Yeah, so 2017, this was BMN 673 was the old name for talazoparib.
Yeah.
So ultimately, at the end of 2018, that drug got approved for patients with BRCA1 and BRCA2 associated triple negative breast cancer. And PARP inhibitors continue to be studied in other groups of patients, not just those patients who have the inherited risk from BRCA1 or 2. But yeah, that was back at that time. So 2018 is when we got those drugs approved, the PARP inhibitors.
Yeah. Okay. Yeah. Second question. What is the one question you would encourage people to ask their doctors when it comes to triple negative, to ask them, their oncologists, about the drugs and treatment?
I mean, I think when I see anyone new with recurrent triple negative breast cancer, I think probably the most important thing for, I think, a patient is really asking the question, are we sure it's triple negative? These tests, we always think about them being black and white, right? It's sort of ER positive or ER negative, HER2 positive or HER2 negative. But the reality is the tests are far from perfect, and there's lots of shades of gray.
Mm-hmm.
So, I just saw a patient this week who initially had ER positive breast cancer and now has metastatic recurrence, and it's triple negative. And so I think, the most important thing for me is it really triple negative? Because if it were ER positive, it opens up these other options for treatment, right? Many other options. And so I think that's probably, one of the most important things to consider getting a second opinion on the pathology to make sure that it isn't actually HER2 positive or ER positive, because that just kind of opens up other treatment options. So I always feel like I like to start with that as probably one of the most critical pieces of information.
Okay.
Mm-hmm.
How long, the next question is, how long can you be on Keytruda? And can it be used on its own without chemo, without the infusion of chemo?
Yeah, good question. So when we first started to look at immunotherapy antibodies like Keytruda in triple negative breast cancer, a lot of the early studies looked at just giving Keytruda alone. And giving it by itself, there are some patients who will have a great response to that. But the number of patients that have a good response, it's overall low. And so over time, we learned that combining Keytruda with chemotherapy was a more effective approach. And so that's why those combination strategies move forward. There was a phase 3 trial that looked at doing chemo versus Keytruda alone.
Mhm.
And the Keytruda was not better than chemotherapy. So that did not move forward, but that was tested in a big trial. And so, the way we have approached it is to give Keytruda in combination with chemotherapy.
Mhm.
And I think that while we start off and give the drugs together, I certainly think if there's a really good response, it's very reasonable to consider dropping the chemotherapy and just staying on Keytruda only as sort of a maintenance treatment. This, of course, is associated with more favorable quality of life and overall experience for patients. And, there are some patients that will have a complete response with immunotherapy. And right now it's recommended that, you continue Keytruda as long as it's working up till 2 years. And if at that point you've been on Keytruda for 2 years and there's no evidence of cancer, you stop the drug and observe. And I've had certainly patients, I just saw in the last month a patient of mine, she's been off Keytruda for 7 years. And she had metastatic breast cancer to the lung and brain. And she has no cancer and is on no treatment.
Wonderful.
So we wish that happened more, but it
Mhm.
is possible with Keytruda that you can get these responses. And I think if we look at all the classes of drugs, it's really immunotherapy that has that greatest potential for getting a result like that. So I have patients in my practice who, amazingly had a great response like that. And they're so wonderful when they happen. But I think we really want more patients to have that. And that's why I'm excited about these new bispecific antibodies, because they appear to be active across all triple negative tumors, not just the 40% of triple negative tumors that are, positive for that PD-L1 marker. So I think we need to improve immunotherapy. We've seen some signs that, it can be curative for some patients. So we need to keep pushing that forward, expand it, for more patients and make the immune therapy approaches better.
Okay.
Yeah.
Next is the VEG targeted study.
Mhm.
Um, is that just for early treatment of triple negative breast cancer?
Yeah, so these trials that are looking at these, it's
Mhm.
kind of like a twist on Keytruda. So they're targeting, the same immune therapy marker, either PD-1 or PD-L1, but then they're targeting this other protein called VEGF. And so these, right now, they're only being studied in recurrent or metastatic triple negative breast cancer. There aren't any early stage studies yet. But they are rapidly moving forward in clinical testing. So the BioNTech drug, the BNT327, that's currently open at about, I think, 30 sites across the US in phase 2 right now. I think by the end of the year, a phase 3 trial will be started. So that's moving very quickly. And then I think the ivonescimab drug, it is approved outside of the US, actually in lung cancer. So I think, that is going to move very quickly forward as well.
That's great.
So it's exciting. I think they're, one of the most exciting new classes of drugs that we have.
Someone asked the question, are there any recommendations on managing side effects from these treatments?
Yeah, I mean, I think as we, you know, um, have done better, right, with new treatments, we've had to contend with lots of new side effects. So with immunotherapy, while generally, patient goes in, receives a Keytruda infusion over 30 minutes, doesn't feel a lot from it, there can be in up to a third of patients some of these immune related side effects. that we need to manage. And so they can, literally be related to inflammation of any organ. So inflammation of the lung, inflammation of the intestinal tract, rash, inflammation of the skin, inflammation of some of the, other glands in the body, like the thyroid gland, the adrenal glands. And so certainly it's important, I think, for patients to be aware of these immune-related side effects, and talk to patients, about, if you have any new diarrhea, shortness of breath, skin rash, right, to let us know. I think the antibody drug conjugates, while they, appear to be working better than chemotherapy for patients with recurrent triple negative breast cancer, I guess my wish would be that they would be better tolerated than chemotherapy. I think for most patients, they really feel the experience from the patient is sort of, it feels like they're on chemotherapy with these drugs. So I think a hope for the future is, continuing to work on improving these antibody drug conjugates and sort of hitting on a drug that, is really effective, but, quite good from a side effect perspective. And so, those are all sort of on my wish list, those things.
Okay. For those of you who are interested, there is a search tool in the chat where you can go and search for clinical trials, and I would encourage you to do that. The question is next, after being on ADC for a year rather, and now being NEAD as stage 4 triple negative breast cancer, what are the maintenance options out there? Is there anything with data behind it?
Yeah, this question is coming up a lot. I mean, it's really coming up a lot in HER2 positive breast cancer, where we're using, these ADCs are moving forward more and more. So if a patient has a great response to an ADC, do they need to just keep staying on it and sort of having to deal with the side effects? I think that ASCENT-04 trial is looking at an ADC with immunotherapy for patients who are whose tumors are PD-L1 positive. And so, I can see in that after a period of time, if the response is good, dropping the antibody drug conjugate and then just continuing the Keytruda. I think right now, though, there isn't a great maintenance. I think some patients are saying, well, maybe can I reduce the frequency rather than coming in and getting this treatment, every 3 weeks? Could I do it every 4 weeks, every 6 weeks? So that is something, I think that's being done, on a patient by patient basis. I think really the only maintenance option that we would have, if the patient is eligible, would be Keytruda. But again, we're waiting on the data of the ADC plus Keytruda data. So it's a tricky problem, and it's hard when you're on a drug and you're having side effects from it if you have a great response, right? So I think probably right now, maybe thinking about reducing the frequency of how often the drug is being given.
Okay. What kind of support do you find patients benefit from the most?
I think, this is a really difficult type of breast cancer to deal with, right? Any recurrent breast cancer, metastatic breast cancer. So certainly, family, all the social, friend, family support is really important. But I think it's important to have, a medical team too, that is there to answer any questions. If you're having symptoms, to respond to those. There's a lot of good data that for patients with recurrent or advanced cancers, that, sort of rapid sort of addressing of symptoms, and better symptom management leads to actually better survival. So I think having, your physician, nurse practitioners, physician assistant, nurses there that you can get through to help you, palliative medicine, right? So good, addressing symptoms in a very timely fashion and doing that effectively, it is important for outcome. And then I think, patient support, right? So like sometimes I have some patients tell me they don't feel like anyone understands, where they are inside. And so sometimes support groups or other sort of patient groups, it can be really a wonderful thing for people to meet other people dealing with the same thing. I think patients feel like they don't need to explain where they are, what's going on, because others who are in it, they just get it. And so I think all of those things are important, but definitely if you're having symptoms from your treatment, from your cancer, addressing those well will lead to a better outcome.
This last question is one that I have specifically. How difficult is it for you to find African Americans to get engaged in your clinical trial so that the outcomes we're getting are addressing the ones that have the highest mortality rate from triple negative?
I think if we look at our clinical trials, right, and how it's done in the United States, right, we have a huge landmass, right, and lots of very different geographies, urban, rural, all over the country. I think, unfortunately, our big cancer centers tend to be clustered in certain locations, and then there are big areas in the country that don't have access to, say, an academic cancer center. And so if we're running a lot of trials through academic cancer centers, then the challenge is that we're not reaching a lot of patients, right, that are out across the country that may be living in states where they don't have an academic cancer center, in more rural places. And so I think we really need to invest in having clinical trials available at cancer clinics, smaller cancer clinics across the country. And I think there is a push in that direction. Certainly, the National Cancer Institute has something called the NCORP system, which is a national oncology community research program. But we need more, community sites and hospitals, clinics to participate in the NCORP clinical trials. So, it's hard if it's a small clinic and it's not a big cancer center. There's not all the infrastructure to run clinical trials. So I think that, there needs to be better support, financial support, staff support for some of these smaller centers across the country, particularly, in areas where patients are not currently able to access a big clinical cancer center. So it is an issue. I think that, the FDA is shining light on it, this issue, that, it's a requirement that companies who are testing a new drug that they're developing, it is a requirement that they have enrollment of a patient population that really reflects the population across the country. And so I think that some of these moves by the FDA are now sort of forcing pharmaceutical companies to look at cancer, clinics and hospitals that they haven't previously looked at, to offer their clinical trials at those locations. So I think some of these, the FDA regulations, these newer regulations, are sort of, making the drug manufacturers, making them ensure that they have a representative population of patients in their trial. So I think it's good for patients overall.
Dr. Telli, I think we're about out of time. Casey, do we have time for other questions, or are we at our deadline?
Unfortunately, we are at our deadline. I know we've had some good conversation today. I hate to cut it off.
Yeah, but it's really been enlightening. So enlightening. Thank you again, Dr. Telli.
Yeah, thanks everyone for joining. And if there are other questions, I'd be happy to field them offline.
I'm just going to wrap us up here by doing a quick thank you to Dr. Telli and Miss Linda. Both were fabulous. The presentation was incredible, and Miss Linda did amazing for fielding those questions and making sure we got through as many as possible. The session today was recorded and is going to be posted to our landing page at komen.org/MBC series, as well as our YouTube page. And as long as you're registered, you will get the link directly to your email address. I do have to offer an apology to our attendees. We noticed that something was a little off in the beginning, but we weren't sure what was going on. And we did figure it out. Unfortunately, there was something wrong with the Zoom timing. And so a lot of our attendees hopped on at the very end thinking it started at 2 p.m. Central and at 1 p.m. Central. I'm not quite sure how that happened. It's the first time in 3 years of doing this that has happened. But don't worry, I'm going to figure it out so it doesn't happen again. I just have to offer you my apologies. I don't know whether it was user error or tech error, but I'm going to figure it out for y'all. Either way, the recording is going to be sent directly to your email so you can rewatch and learn from Dr. Telli and hear the conversations that we had. Again, I'm so sorry. It's never happened before. I had no intention of it happening. But I also just need to quickly thank our sponsors. They're why we're able to put this great content on for you guys month in and month out. So thank you to AstraZeneca, Merck and Gilead, and thank you to Daiichi Sankyo as well. I did drop a survey in the link, in the chat. Now, of course, if you weren't able to join us live, please wait until you get the recording and then provide your feedback. But part of the survey is also what topics you want to hear about, who you want to hear from, what will best support you through this metastatic breast cancer journey. And that's really important for me when I'm planning all this programming out. So please don't be shy. I want to hear from you. It's how we make sure this is valuable content. So finally, lastly, our next MBC Impact Series event is March 10th, so just a couple weeks away, and is focusing on HER2-low metastatic breast cancer, which we touched on briefly today, but we'll do a much bigger deep dive next month. So thank you again for joining us. Again, I offer my sincere apologies, guys. I'm not sure what happened, but we'll figure it out and make sure it doesn't happen again. Stay safe, stay connected, and hopefully I'll see you next time.
Thank you.
Thank you.